High blood LDL cholesterol is widely accepted to be a major risk factor for CHD, which is the leading cause of morbidity and mortality around the globe. As part of a normal metabolism, the liver releases cholesterol into the bloodstream. A member of the lipids family and an essential molecular building block in the human body, cholesterol circulates around the bloodstream attached to proteins, forming complexes known as low-density lipoprotein cholesterol, or LDL. In this form, cholesterol is carried to the tissues that need it. However, too much LDL can be problematic, as it can lead to the formation and build-up of atherosclerotic plaques on artery walls. This can cause problems such as obstructions, hypertension, or coronary heart disease. Cholesterol present in the gut is contained within bile from the liver and from food sources in the diet. In order to be absorbed from the gut into the bloodstream, cholesterol mixes with bile salts, fatty acids, lecithin, and triglycerides to form tiny water-soluble packets called dietary mix micelles. The micelles then make contact with the intestinal wall, where their contents are taken up into the cell. Once inside the cell, the micelle contents are repackaged into spherical particles called chylomicrons. The chylomicrons then carry their fatty contents into the bloodstream, where they are delivered to their target tissues, including the liver. Newly synthesized cholesterol and cholesterol which has been absorbed from the gut is then released from the liver into the bloodstream in the form of lipoproteins, which gradually become LDL cholesterol. However, there is an ingredient called plant sterols which can interfere with the absorption of cholesterol from the gut and significantly reduce the amount of cholesterol that reaches the bloodstream. The exact details of how they do this are not fully understood, but there is strong evidence to suggest that plant sterols displace cholesterol from the micelles formed in the gut, which are essential for cholesterol absorption. This reduces the amount of cholesterol in the micelles and thus the amount that is presented to the lining of the gut wall in a form that can be absorbed. The cholesterol that is not incorporated into micelles is then subsequently excreted from the body. The small amount of plant sterols that are taken up into the cells lining the small intestine are then actively pumped back into the gut lumen and excreted along with cholesterol. A wealth of scientific evidence clearly indicates that a daily intake of 2 to 2.5 grams of plant sterols per day leads to a reduction in intestinal cholesterol absorption of 30 to 40% and an average of 10% reduction in blood LDL cholesterol levels. This 10% reduction is important because evidence suggests it can cut the risk of CHD by up to 20%.